from University of Limerick, Thailand.
Yes. I will share my screen. Let me see. You are able? Can you see my screen?
Yes.
Okay, I will.
Full screen. Okay.
Yeah, can you see?
It's okay.
Okay, perfect. Okay, so good morning, everyone. My talk will be on the impact of BILF1, a G protein coupled receptor encoded by Epstein Barr virus in lymphomagenesis and its therapeutic potential. So, Epstein Barr virus, known also as EBV, it's a gamma herpes virus which infects more than 90% of people worldwide. Since its discovery in 1964, the virus has been linked to many human malignancy, which can be divided in two main groups. Malignancy with an epithelial origin, such as nasopharyngeal carcinoma, gastric cancer, and tumors with a lymphoid origin, like Hodgkin lymphoma, Burkitt lymphoma. EBV is transmitted through saliva, and after primary infection, the virus will establish its latency in B cells, which represent the reservoir of the virus. In this state, EBV will express a low number of viral proteins. In contrast, when the virus enters in a lytic phase, which led him to infect other cells and thus other people, EBV expresses the highest number of viral proteins, more than 80. The protein called BILF1 is a transactivator gene which switches from latent to lytic phase. Based on the expression of these viral proteins, we have different types of latency, called latency type 1, 2, and 3. Latency type 1 is characterized by the presence of just one protein, called EBNA1, along with long and small non-coding RNAs. We can find this type of latency in Burkitt lymphoma. Burkitt lymphoma is the most common tumors in children in Africa. Burkitt is a form of non-Hodgkin lymphoma, and the progenitor of this tumor are the B cells. The tumor can be divided into three main clinical variants: sporadic, which occurs worldwide; endemic, which occurs predominantly in sub-Saharan Africa; and immunodeficiency associated BILF, Burkitt lymphoma, which occurs mainly in the setting of HIV. The association with EBV is highly variable, with more than 90% of the endemic cases diagnosed as EBV. As previously mentioned, the virus expresses a latency type 1 in Burkitt. However, for the first time in our paper published in PLOS Pathogens by Abate et al. In 2015, we showed that Burkitt lymphomas present a non-canonical latency characterized by the expression of other viral proteins like LP1, LP2, BZLF1, and BILF1. We focus our attention on BILF1 mainly because this gene is an active viral G protein coupled receptor, or GPCR, and we know that GPCRs are the most successful class of drug target for the treatments of human disorders today. This takes more importance if we consider that although EBV is a problem of worldwide importance, no vaccines or antiviral drugs have been yet approved. What we know from literature is that BILF1 is involved in immune evasion from T-cell recognition, and it has a transforming ability in mice. However, discordant results regarding its expression are reported in literature. Some authors found these genes in both latent and lytic phases. Others observed the presence of BILF1 in the lytic program only. So based on this, we wondered if BILF1 is expressed in Burkitt lymphoma and in which phase we can appreciate its expression. To reply this question, we reanalyzed data from our RNA-seq performed in Barkett's cases published in 2015, and we also used some of these cases to check the expression of BILF1 by applying three-time PCR. As indicated by blue arrows, BILF1 was expressed in almost all cases. We also confirmed the presence of BILF1 by using RNA-scope assay. RNA-scope assay is an in situ hybridization technique which led us to detect the presence of BILF1 mRNA. The characteristic staining of RNA-scope consists in a brown punctate dots, as you can see in the top, BILF1, and in bottom right, internal positive control. And you can also appreciate some EBV positive Barkett cell lines used as positive controls, which show the presence of BILF1. So after confirming that BILF1 is expressed in Barkett's lymphoma, we aim to define the phase in which BILF1 is expressed. To accomplish that, we performed a double staining detecting BZLF1 by immunohistochemistry and BILF1 by RNA-scope. We found that in some Barkett's cases, BILF1 was expressed along with BZLF1 expression, right in red, whereas other Barkett's cases express BILF1 and not BZLF1, left. Altogether, this data suggests BILF1 as a latent gene. In addition, for the first time, we were able to design and optimize in immunohistochemistry an antibody against BILF1. This led us for the first time to appreciate the presence of BILF1 protein, which localized in the membranes of two EBV negative Barkett cell lines transfected with BILF1 plasmid bottom in respect to the parental cell lines used as a negative control top. So we applied these antibodies to stain additional Barkett's cases characterized by the presence of both BZF1 and BILF1. The staining of BILF1 protein was also compared against the expression of BILF1 mRNA detected by digital PCR, as you can see in these graphs. After this, we aim to define the contribution of BILF1 to the transformation of B cells, the progenitor of Barkett's lymphoma. To do so, we isolated germinal center B cells from fresh tonsils. We transfected these cells using BILF1 plasmid, and we collected these cells 72 hours after transfection to perform an RNA-seq assay. We compared this data against the data from RNA-seq previously performed in Barkett's primary tumors, and we found that genes upregulated by BILF1 in germinal center B cells were significantly enriched among those genes that were upregulated by BILF1 in Barkett's cases expressing BILF1. Top. Also, genes downregulated in germinal center B cells by BILF1 were significantly overlapping the genes downregulated by BILF1 in primary tumors. Bottom. So, to understand the biological impact of BILF1 in Barkett's cases, we performed a pathway analysis of these dysregulated genes. As lighted in red in the table, we found an enrichment of genes involved in cell proliferation, DNA repair, AKT-PI3K, and M and MYC pathway, which represent the hallmarks of Barkett's lymphoma. So we selected some of these dysregulated genes, and we confirmed their change in germinal center B cells after BILF1 transfection in gray, and in Barkett's primary tumor in blue. So in the end, we explored the impact of BILF1 on the phenotype of Barkett cell lines. So to investigate this aspect, we select two EBV negative Barkett cell lines. We transfected the BILF1 plasmid, and we performed an MTS assay to check the rate of cell proliferation. As you can see from the picture, we can appreciate an increase of cell proliferation after 48 hours after transfection of BILF1, and this change was stronger and marked after 72 hours. So based on these results, we aim to confirm the positive relationship between BIF1 and AKT P3K pathway. To accomplish that, we transfect two ABV negative bucket cell lines with BILF1 plasmid, and we performed an MTS assay. And in parallel, we use some of these cells to make cytospin and stain the cells using an antibody against phospho-AKT. As you can see from the image, we found a strong activation of AKT-P3K pathway in cell transfected with BIF1 bottom in respect to their parental cell line top, which express a basal level of AKT-P3K pathway. In conclusion, we can say that BIF1 is a latent gene. BIF1 drives a bucket-like transcription program involving AKT-P3K pathway and MYC pathway when expressing primary human germinal center B cells, and BIF1 could be a realistic therapeutic target. So, thank you for your attention. Please let me to express my gratitude to my supervisor, Professor Paul Murray, who guided me in these significant results, and my colleagues who helped me with experiments. I would like to express my gratitude also to the European Committee, who under Horizon 2020 and the Marie Curie Fellow funded my grant. Thanks a lot to everyone for listening, and also many thanks and a big thanks goes to the organizing committee for giving me the possibility to be a speaker in this international and interesting webinar on international cancer and breast cancer therapy. Thanks a lot.
Thank you, Dr. Lucia.
Thank you.
For your excellent presentation.
Thanks.
Our participants enjoy it.
Thanks a lot.
Now, we are waiting for your question. If you have anyone, participants. Question to ask Dr. Lucia. They seem to have got everything, doctor.